Hi, I'm Andrea. Welcome to today's Harper Lecture. The Harper Lecture series highlights the University of Chicago's collective commitment to the highest aspirations and standards in research and education. Today, you will hear from our world-class faculty and enjoy stimulating conversations on critical topics, all in the comfort of your home.
We extend our appreciation in advance to our global university community for their collective efforts in supporting the mission of the university in today's deeply challenging environment. Technology has allowed us to remain connected and engaged, and tonight, you too will be part of the conversations. A couple of notes. All participants will be muted. However, we do encourage you to ask questions. We will have plenty of time for questions at the end of the presentation. If you have issues with audio, you may want to shut down your programs running in the background or dial in from your phone.
Hi, I'm Colin. On behalf of everyone at the UChicago Alumni Office, we offer our deepest gratitude and appreciation for your continued support. We're proud to be part of this community as it has come together during these trying times to invest in our students, empower our faculty and scientists, and extend care and connection. And now it's my pleasure to welcome you to the 41st season of the University of Chicago's Harper Lecture Series. Thank you for being with us. Enjoy.
Hi, everyone. Welcome to today's Harper Lecture, Industrial Scale Brain Mapping with Professor Bobby Kasthuri. I'm Andrea Hodgman, Associate Director for Intellectual Engagement and Travel and the Program Manager for the Harper Lecture Series. I'm so pleased to welcome you to today's event. On behalf of everyone at the UChicago Alumni Office, thank you for joining us. Please allow me to introduce our moderator. Peter Littlewood is a professor in the Department of Physics, James Franck Institute, and the college, and a fellow at the Pritzker School of Molecular Engineering. He came to UChicago in 2011 as Associate Lab Director and then Lab Director at Argonne National Lab, returning full-time to the university in 2017. His particular areas of current focus include connectomics, measuring and modeling the connectivity of every neuron in a brain, non-equilibrium phase transitions in strongly coupled light matter systems, and materials, technology, and policy for energy. Professors Kasthuri and Littlewood, thank you for joining us. Professor Littlewood, I'll turn things over to you.
Thank you very much, Andrea. So good evening, as Andrea Littlewood. I'm Professor of Physics at the university, and I'm speaking to you from the Harper Library that you might remember. I should preface my introduction of Bobby by explaining why it is that a theoretical physicist has the honor to introduce a talk by an accomplished neuroscientist. A few years ago, I was director of Argonne. Argonne is a Department of Energy national laboratory that's in the suburbs of Chicago and is managed by the University of Chicago as it has been since 1946. Argonne engages in big science projects. It has one of the world's largest computers, an X-ray microscope that is over 1 kilometer in circumference, and is a center for US battery science, amongst many, many projects. But when I came across a researcher who had the bold vision that he wanted to measure all of the connections between every neuron in a mammalian brain, I was immediately taken by that idea. So to paraphrase Victor Kiam commercial for a brand of shaver, I liked the idea so much I bought the company. So I set out to recruit tonight's speaker to UChicago and to Argonne, and I'm delighted to be able to introduce him tonight. So Bobby Kasthuri is an assistant professor in the Department of Neurobiology at the University of Chicago. He earned his medical degree from Washington University School of Medicine and a DPhil from Oxford University, where he studied as a Rhodes Scholar. As a postdoctoral fellow, he developed an automated approach to large volume serial microscopy, connectomics, which he will explain. And his lab continues to innovate new approaches to electron microscopic-based reconstructions of neural systems, including making samples more amenable to automatic segmentation and combining proteomic and genomic approaches with electron microscopy. The lab is also exploring the use of high-energy X-rays from synchrotron sources for mapping brains in their entirety. And the lab, of course, is applying these techniques to developing adult and aged brains in service of answering the question, how do brains grow up and age? Dr. Kasthuri, the floor is yours.
First, I want to thank Professor Littlewood Peter for that very nice introduction. I'd like to thank the organizers of the Harper Lecture for inviting me. It's really a great pleasure and a great honor to be able to give this lecture and to tell you both what my lab is working on and what a large, I'm hoping, growing part of neuroscience is working on. As Peter may have mentioned, I'm an assistant professor in the Department of Neurobiology and a neuroscientist at Argonne National Labs. That's sort of like saying I've just kind of started in this job as being a professor and running a lab. So I thought one way that I would start my lecture was to share with you one of the, I think one of the key things I learned about being a professor from my postdoctoral advisor. And he said, and I believe this is true not just for professors, but for professionals in general, that the key part of the word is to profess something, to stand for something. Beyond just our ability to collect facts and relay them, what is our opinion? What is our worldview about science and how it should be done? So in addition to telling you about the work that we do, I thought I would also share with you a little bit of my worldviews and my philosophies. And just to start, one of the, I think, places where my worldview and the philosophy of the University of Chicago, I was quite surprised to find, are really in union, is that we should really be thinking about the big ideas and the big things that we can contribute to science. To give you an idea of what I mean by that, I thought it would be useful to sort of summarize what I think is probably the greatest sort of scientific and intellectual achievement of the 20th century, which I'll show you in the next slide. And to me, I think the great achievement of the 20th century is really mapping the genetic basis of life, basically the discovery of heredity and DNA and how the two play with each other. Interestingly for me, that these discoveries of DNA are actually probably about maps, particularly two maps of DNA that span the 20th century. The first done on the left-hand side by three scientists, Watson, Crick, and Rosalind Franklin, who used X-rays to map how individual base pairs, individual pieces of DNA fit together. And then the other great map about DNA, which happened several decades later, was the map of the human genome, which was published in 1990. And here, this was a map at a much larger scale of how every gene and non-gene coding sequence in a piece of human DNA was mapped relative to each other. What I find interesting about these projects is that they didn't have a particular hypothesis. They didn't go out to test a particular idea, but they were large data sets taken about a particular part of our body, our genetic material, which had never been taken before, and therefore enabled an entire world of science to come afterwards. If this was the great achievement of the 20th century, my suspicion is that the great achievement of the 21st century will be another map, but this map not of DNA, but of the physical basis of thoughts and how our brains work. Here, for example, is a picture or a depiction of a human brain, about 3 pounds, about fits into the 2 palms cupped together. But one could argue that much of who we are, much of what we can achieve is found in our brains. And just to give you an idea of what a map of a brain would look like, I thought we could start with some of the very first maps of brains ever made by this gentleman, Santiago Ramon y Cajal, in the early 20th century. And you'll see Santiago here with the microscope. This is another view of mine that technology is what drives neuroscience. And what you'll see on the right-hand side is one of the very first pictures of a brain. And I thought I would just go through it together so we're on the same page. Santiago Cajal was one of the first people to realize that the brain was made of individual cells. Here you can see labeled as G, A, and C, and one that's not labeled off on the side. They're often called neurons. That was Cajal's first big discovery. His second big discovery is that neurons, unlike many other cells in your body, are polarized. So for example, a skin cell or a liver cell, they're very symmetrical. They look the same from whatever angle you look at them. But neurons are slightly different. In this particular picture of neuron A, you can see a large tree that extends to the north of the neuron, which we call the dendritic tree, and then a long cable that stretches out from the other side of the neuron, which we call the axon. Now, I'm a little bit of a narcissist, and I imagine that if I had a microscope and a stain, and I was the first person to look in a brain, I might have been able to figure out that neurons have dendrites and axons. But there's a part of this picture that's truly, I think, genius. And if we were in a room together, I would quiz you, but I'll give you the answer. The answer here is that the arrows, which of course aren't found in any brain, unfortunately for neuroscientists, and what Cahal is showing with these arrows is the flow of information in the nervous system, at least in mammalian nervous systems, where information or neuronal or electrical activity travels up axons into the dendrites of other neurons, out through their soma or their cell body, into their axon, out into the dendrites of another neuron, and so on and so on and so on. And you may have heard of neural networks or artificial intelligence algorithms that operate on neural networks. This is the very first example of a neural network, a set of neurons that communicate information to each other. And normally I would say, there's a very strong hypothesis in neuroscience that this pattern of connections actually subserves everything that we do, all the sensations that we receive, our decisions, our behaviors. But rather than sort of try to make the argument for you, I thought for the next couple of minutes, I would share with you, a recent result, if you will, in neuroscience that I became aware of, that actually convinced me, and I suspect will convince you further than any arguments that I could make. Now, before I can tell you about this result, I have to just make sure we're on the same page about actually humans and how humans are born. It turns out, and you're probably well aware, that in a small certain percentage of humans, much less than 1%, humans are born conjoined twins, where they're joined physically with another twin. And it turns out that even among conjoined twins, there's a very rare case where twins are actually joined at the head, called cranial phagus twins. And they're about 1 in 2.5 million births. And the vast majority of them do not survive 24 hours. And I'd like to tell you the story about one of these cases. This is the case of Krista and Tatiana Hogan, who were born about 15 years ago in Vancouver, British Columbia. And soon after they were born, a scan of their brain revealed that they had two brains joined together in a particular area of the brain called the thalamus, which our lab actually studies along with our chairman, one of the fathers of the thalamus, Murray Sherman. And like many children, they confounded their expectations, and they lived well past 24 hours. And they're now at the age, I think about 15, getting ready to start high school. And they represent the kind of remarkable discovery, which is that as they are actually physically linked by connections in their brains, their sensations and their thoughts are also linked. And I won't spend too much time on this. If people are interested, I put the documentary that I saw very recently and some other references to follow up. But here in these panels, you can see this sort of effect of physically linking connections. Here, basically, in the panels on the right, a mom and big brother can close the eyes of one of the twins and touch the other twins on some location, her cheek. And the twin that's blinded is able to respond, her cheek. She can touch her on the knee and say, her knee. In another case, the twin can have her eyes closed. And another twin can be shown, for example, I hope you can see a pink pig. And the twin who cannot see can be asked, what color? And she responds, pink. And in fact, I was discussing this very result with my chairman Murray earlier today. And I realized what is remarkable about this, other than the remarkable courage of these twins. And I think what is remarkable is this is exactly the result you would expect based on our current understanding of how neurons connect with each other, and that those connections are the basis of much of who we are and what we do, including sensation. So with this sort of little vignette, I hope I could convince you that it's worth mapping the brain and understanding these connections. And I suspect some of you are asking, reasonably so, okay, well, why hasn't it done yet? And part of the reason it's not done yet is that mapping the brain is really hard, because in human brains, like the brains, like your brains actually, we suspect that there aren't 4 neurons like we saw in the Cahal diagram, but something like 100 billion neurons. And each of them make on order 10,000 connections with each other. So if you do that multiplication together, you get a number like a quadrillion connections in a human brain. That's a hard number for me to understand. One way to think about that is that there's that's an order of magnitude more than the stars in the Milky Way galaxy. Now, that's a large number, but you may have already heard that people are actually trying to map all the stars in the Milky Way galaxy. Well, there's one special difference about mapping brains that I'd like to talk to you about, so I can tell you a little bit about the technology we've done to do it. Now, one way to conceptualize this is imagine that if a connection between two neurons was maybe the size of the grain of sand, like you see here, relative to a hand and a finger. And if I collected a quadrillion of those connections that were each the size of the grain of sand, I would soon get to buildings, to a structure that was probably more massive than many of the buildings in downtown Chicago. Now, our brains don't have the volume of a city. They actually have a much smaller volume, about 1.3 quarts, 1 1/3 of a milk quart bottle, if anyone remembers. Now, then to, of course, squeeze 4 trillion connections into that volume means a few things. One, each of those connections, or a synapse, is so small that it's actually smaller than the wavelength of visible light. Second, neurons receive thousands of connections and make thousands of connections. Third, neurons actually travel from one side of the brain to the other side of the brain, from one side to the other. And as a result, this web, or connectome, as we call it, is unbelievably complicated. Perhaps one of the most complicated things ever developed. So complicated that we realized that the only way to map it was to use very high energy electrons. And I'm just going to spend 1 minute telling you about a sort of serendipitous invention that we made, which allowed us to map brains at scale. So when I was a postdoc at Harvard, one of the things we developed was a way to take a piece of brain, prepare it for electron microscopy, and by creating an automated taping mechanism, collect thousands of sections of the same brain in series. Now, remember, each of these sections is only about 300 atoms thick, so the sections don't change very much from one to the other, but they give us enough resolution that we can use an electron microscope, take a picture of each of those sections, and then ultimately reconstruct the brain digitally. I'll just spend about a minute showing you what these EM data sets look like so you can get a kind of idea of the complexity of the brain. So here is a picture of a mouse cortex, similar to Cahal's picture. You might notice in the middle a few neurons that I'm going to zoom up on. Here they are again. 1, 2, 3, 4, 5, and 6. Now, unlike Cahal, where there was all empty space between these neurons, we can actually see the processes of neurons that are filling it. So I'm going to zoom up some more, and then I'm going to zoom up one more. And right in the middle of your screen is a profile. Remember, this is a section of a brain of an oval filled with circles. And actually in 3D, these are called synaptic vesicles. And the profile of the neuron just above it is where this neuron, this axon, if you remember from Cahal, makes a synapse with this dendrite. That's one. There are about 100 billion more in the mouse brain that I'd like to map. Now, just to tell you how we go from this to a 3-dimensional model, in this movie, what you'll see is a stack of electron microscopic images of volume. And in one of these images, I can trace a single neuron and from section to section find the same neuron and ultimately convert it into a 3-dimensional model. At this particular neuron, this dendrite that you see in orange is actually receiving a single connection from an axon that you see in blue, as you can, is typified by these synaptic vesicles. For my postdoctoral project, I actually decided to reconstruct an extremely small part of the brain, smaller than a grain of sand, just to get an idea of how complicated a mouse brain could be in this extremely small volume. And in this explosion that you see of that extremely small volume, you'll see the reconstructions of thousands of axons and hundreds of dendrites from different neurons that seem to make this huge mess. And yet we know that there are rules that the brain must follow to establish these connections, because the system works again and again and again. To give you an idea of how small this volume is and how complicated it is, here I've just sort of zoomed out again and again to give you an idea. So here at the top left is that explosion. That explosion came from a very small volume relative to neurons that you might have seen, remember from Cahal. And all of these neurons occupy even a very small fraction of the entire mouse brain. So you might imagine that this is doing a whole mouse brain could be an incredible task, but in fact, this task is already being discussed. In fact, in a paper published with at least 3 University of Chicago professors, including Peter, we propose to map the largest single data set in the history of biology, which is the mouse connectome, where a human genome would have been about 200 gigabytes. The mouse connectome, we believe, will be 2 billion. And the reason we're quite confident is because we have a resource that we think is perfectly suited for it. Peter already mentioned it, which is Argonne National Labs. Because of this particular and special relationship between Chicago and Argonne, our lab and hopefully soon many other neuroscientists are able to leverage scientific tools that you cannot find anywhere else, including synchrotron X-rays, including electron microscopes, and most importantly, including some of the largest computers on the planet to help us analyze these data sets. So great. So it feels like we might be able to get the technology, but what specific question am I interested in? In the last 15 minutes of this lecture, I thought if I could have my druthers and use this technology to understand one thing about the brain, I think it would be this question. And that is, how do human brains grow up? As an example, I'm showing you 3, of course, very famous humans: Jane Goodall, Mahatma Gandhi, and Albert Einstein, from left to right. And although you may have seen their pictures, you may not have seen their baby pictures above them. And what I'm interested in is how is that process from growing from child to adult happen? How do we develop these worldviews and make these tremendous discoveries? It's important to say, although genes are probably involved, as important to be involved are what they learned over the course of their lifetimes to make their brains. As soon as I started investigating this, of course, I found another University of Chicago connection. And this is the work of Peter Huttenlocher, who was a pediatric neurologist here at university, who spent a large fraction of his time thinking about how human brains grow up. And in fact, his wife was also a professor here. He was a professor here for 30 years, and his wife was a professor in the psychology department. And what Huttenlocher hypothesized, proposed, is interestingly the way human brains develop. Here you'll see one of his very famous graphs where you see human age on the x-axis and the number of connections on the y-axis. The number of connections actually goes down as a function. There's actually pruning in it. Now, what Huttenlocher wasn't able to get to was to see how different this was across species. And as a quick example of what I imagine, I'm showing you a picture from one of my favorite books called Growing Young, which talks about Huttenlocher. But this book makes the argument, I think, summarized in this picture. Here's a picture of a juvenile chimpanzee on top and an adult chimpanzee on the bottom. And what the author of Growing Young, the argument he makes is that the juvenile chimpanzee is much more similar to the adult human than the adult chimpanzee. Basically, the idea is that the chimpanzee is aging, or the chimpanzee's development is accelerated relative to us. And actually, it turns out that this is apparently well known in the arts. And I found a poem by Alexander Pope on the nature of man, which I thought we could read together. And Pope says, "The beast and bird their common charge attend, their mothers nurse it and their sires defend. The young dismissed to wander earth and air, there stops the instinct and there end the care. The link dissolves, each seeks a fresh embrace, another love succeeds, another race." And this is the part that I think has always resonated with me. A longer care man's helpless kind demands, that longer care contracts more lasting bands. That if you were to ask me what it is that makes us special, both in our brains and as humans, it's this, that we take so long to develop relative to other animals, relative to chimpanzees, for example. And it's this protracted development that allows us to learn from the environment around us. And one of the things that the lab is interested in is characterizing that, is actually seeing whether we can find similar Huttenlocher curves across species and across different brain regions. Because of course, not all parts of the brain develop at the same time. And in fact, in humans, and in mammals in general, there's a broad idea that the brain develops from the back to the front, that the very earliest parts of our brain, of our processing, for example, of a visual stream, which happen in the very back of the brain, develop first. And then as you move further and you become more abstracted, when you're not only looking at edges or colors, but you're grouping things into faces or objects, that this kind of processing happens further up towards the front of our brain, and that those processes are delayed potentially relative to the beginning of the brain. The reason I'm interested in this is because if we can understand this process, I suspect this is the way that we'll not only be able to understand what it means to be a human, but I think there are several other payoffs as a result. And so for the last few minutes of this presentation, I thought I could tell you what those particular payoffs are. Number one of the very last samples I collected as a postdoc in Jeff Lichtman's lab at Harvard was actually a sample of from a human being who had undergone epilepsy and had a resection as part of their therapy. And as a result, we were able to obtain that tissue and actually for the first time now do a large resolution map of the actual human brain and how it connects here in collaboration with the Lichtman lab. What you can see is a piece of human temporal cortex, particularly in this patient that was slightly adjacent to the area that was epileptic. And if you zoom in, if you look closely, each of those little things is a neuron and its dendrite and its axon for the first time ever mapped in a human being of 100,000 different neurons and their connections to ultimately compare. I bring this up because not only is this work happening at Harvard, but for example, a very specific application is here with Peter Warnke, who's a professor in the Department of Neurosurgery. We're also taking new biopsies, but in this case, we're taking biopsies of patients with a particular disease called Parkinson's, a movement disease. And one of the treatments, current state-of-the-art treatments for Parkinson's, is to put a stimulating electrode inside the brain to stimulate it. And Peter is one of the world's experts in it. But interestingly, some patients will come in and respond well to the stimulation, and some patients will come in and not respond well. One of the hypotheses is that patients that don't respond well, it's because the connections in that particular part of the brain that's affected by Parkinson's, the substantia nigra, have been decimated. So with Peter, who's a world's expert in collecting biopsy samples, we're piloting some studies to see whether we can use connectomic reconstructions to predict how these patients will behave. And if successful, whether there are other things about the brain that will allow us to predict future neurodegenerative diseases. This is just one example, I think, of what the payoffs could be. A separate example, slightly related to that, is that there's a growing kind of consensus that several types of mental disorders are actually what we call connectopies, or neurons that are miswired in brains, causing pathological result. And there's a very specific version of this because I think often about the development of the brain and what it means. And I'm hoping to share this story with you. When I was a postdoctoral scholar, my daughter, who I think at the time was about 4 years old, turned out that she had an imaginary friend. And later I learned from my mother that I also had an imaginary friend. And I became interested in this, and I began to actually kind of quantify her, quantify the imaginary friend the same way that psychiatrists in the hospital quantify, for example, schizophrenia, because it seems similar to me. Imaginary voices, this depth of belief. And at one point, I actually presented her as a potential patient at Harvard. And one thing that shocked me is that the psychiatrists and the physicians there couldn't tell the difference between my daughter as imaginary friend and perhaps a paranoid adult schizophrenic hearing auditory or visual hallucinations. And then the more I thought about it, my wife, who told me when she was young, she had a sort of obsessive compulsive disorder where she had to watch the same video and hear the same story again and again and again. Now, again, this was perfectly normal behavior in her as a child. But if I told you that my postdoc had obsessive compulsive disorder and needed to watch the same movie every day, we might have a different conclusion. And it started to occur to me that one of the differences that there are lots of behaviors in children that are considered perfectly appropriate, but those same behaviors, imaginary friends, obsessive compulsively listening to the same story, in adults are considered aberrant. And I started to think, well, is it possible that some of what we call mental illness are just Peter Pan brains? Brains that refuse to grow up, brains that where other brains continue to change their circuits and change their connections. These brains keep their connections of childhood, therefore displaying some of the behaviors of childhood, therefore perhaps being labeled as pathological as adults. One direct result of this could be that when we start to compare wiring diagrams of young animals, old animals, and perhaps schizophrenic animals, that the wiring diagrams of young animal brains and schizophrenic brains are more likely to be common than schizophrenic brains and adult brains. Of course, just a potential hypothesis, but this is the sort of avenue for which I think this kind of technology could be applied. Something else that I think is very interesting in these approaches is the idea that we can reverse engineer many of the things that human brains do into algorithms. I know that we often hear about how algorithms are getting smarter every day. They can beat us in chess and Go. Apparently they can beat us in poker now, which is always more disturbing, which means they're better at us at bluffing. But I would argue, and I would say, none of those algorithms can actually move with the grace of a human being, move any of those chess pieces with the grace of a human being, can see and recognize the chessboard, walk towards it, sit down and play the game like we can. So I think there's many things, many inspirations that the brain has left to teach us. And I think this is one example that we will have possibilities of reverse engineering, for example, the way animals move into our algorithms and our robots. The other payoff that I think could be tremendous is really from working with Peter in a DOE lab. One thing which perhaps many people know is that most brains, human brains, operate on about 20 watts of energy. And now these light bulbs, including the light bulb in my bedroom, are maybe 60 watts of energy. And so, and many of you out there may have thought of your friends or your colleagues or your family as dim bulbs. And of course, you're right. But it's not the fact that they're dim and it's what they can do with 20 watts of energy. Peter has told me many times that in the next 20 years, the largest part of our energy budget will not be the oil that we put in our cars or in our airplanes, but it will be all the images and the movies and the information that we constantly move up and down the internet. Somehow the brain has discovered to do it, how to do that for 20 watts of energy, and I hope that mapping it will help us understand this process better. Just for the last couple of slides for this presentation, I also wanted to end it thinking with some larger questions, particularly about how science is done and the benefits of these large projects. I think the best way for me to do that is to just read to you one of my favorite quotes, perhaps of all time. This is the quote by President Kennedy at Rice University in 1962, when he announces that we go to the moon. And I'll just read it to you because I love how presidents talk like this. And we choose to go to the moon in this decade and do the other things, not because they're easy, but because they're hard. But because that goal will organize and measure the best of our energies, because that challenge is one we are willing to accept, unwilling to postpone, and one which we intend to win. But why, some say, the moon? Why choose this as our goal? They may as well ask, why climb the highest mountain? Why 35 years ago fly the Atlantic? Why does Rice play Texas? One additional piece that's not in this quote that I find amazing is that I then discovered that in 1969, when we land on the moon, the average age of a NASA scientist is 28 years old. That means, and I don't know what the average age of a NASA scientist is now, but I suspect it's a lot older than 28 years old. That means those 28-year-olds, when Kennedy announces 6 years earlier, they're in college. and something inspirational happens with this large science. And that going to the moon has fundamentally changed us, not just for the moon, but by what it provided for the rest of the society. And I'd like to end this talk with saying, you know, it is amazing to me that that I was able to come to a place like the University of Chicago, that has kind of the resources and the kind of the mentality to actually imagine science happening at this scale. As Peter mentioned, none of this would really be happening without a very strong collaboration with Argonne National Labs. But as you may have heard, and I'd be happy to discuss, we have many collaborations with the UChicago Medical Hospital, with collaborations at the MBL for looking at exotic animals that Peter and others are interested in, like the octopus. And because of this, we realized that there's a real opportunity here at the university to go beyond just perhaps a particular wiring diagram or a particular connectome, but to actually set up the resources to enable democratic access, to really do a large contribution, which isn't just do our science, but set up the infrastructure and the access to let everyone do their science. And sorry, I can't help myself with one last story, just because we talk about rocket ships. This is Robert Goddard, Goddard Space Center, the aerospace engineer the first person to launch a rocket, maybe 300 feet in the air. And when Goddard launched his rocket, he immediately wrote in his lab notebook, "We're ready to go to Mars." And I believe this was in 1916. And I just love this story because the New York Times found out about it. And in a editorial, they wrote, "Professor Goddard does not know the relation between action and reaction, and the need to have something better than a vacuum against which to react. He seems to lack the basic knowledge ladled out daily in high schools." And it took the Times, I think, 40 years after we landed on the moon to publish a retraction, and in which they said, "Further investigation and experimentation have confirmed the findings of Isaac Newton in the 17th century, and it is now definitely established that a rocket can function in a vacuum." I love this story because of this idea that the entire scientific establishment and the federal government refused to fund Goddard. And it was a private philanthropy, actually, that pushed him to finish his experiments and is the reason that we have NASA today. And in the last 30 seconds, I just want to put up all the people in the lab that have actually done all the work. Sean, Vandana, Greg, our postdoctoral fellows who work with me, Hanyu, Stas, and Dawn are the graduate students who provided all of this data. We have large collaborations with Argonne, both the X-ray group and the supercomputing group, and a particular thanks to Peter for taking a chance and hiring me all those years ago. Thank you very much. I believe Andrew is coming back, and I would love to take questions.
So Bobby, thank you so much. That was tremendous, and I think I got it right. I'm still waiting. So the first thing I just want to remind people is that there is a Q&A. So please put your questions into the Q&A. We have several already. We will certainly do our best to answer all of them. We may not be able to answer all of them as they come in, but we will certainly get back to you. So I like the first question, right? So there's an undergrad says, "Do we have opportunities to join this research group as student research assistants?" Thanks. That was what I said, right? So there's lots to do, and there are many people here. So let me head in a slightly different direction, which is ethics. What do you think are the ethical issues associated with the kind of research we're now doing with brains?
So I think it is undeniable that there will be ethical issues associated with what we're doing. We will be discovering how brains work. We will be manipulating brains. It will give us the capability to manipulate those brains. It will give us the capability to learn things about people, potentially, if we could do this, that they might not want. And I think this is and luckily, I think there's a separate path in neuroscience and in the Brain Initiative, et cetera, thinking about ethics. But I asked this question to you, Peter. I know that there are ethical considerations that make it hard for me. I remember a similar question being asked to Robert Oppenheimer about the bomb and about physicists in general studying these fundamental properties of physics when there's always the possibility that it could be used in unethical fashion. I think Oppenheimer's answer, but please correct me, is that I just can't help it. I'm curious about how it works. And although I know that there are ethical concerns that should be we should care about and monitor, I just can't help it. I have to know how these wiring diagrams work.
Yeah, I mean, I think look, as we understand from the early days of thinking about nuclear physics, the ethical concerns that turn up later are things that you never really imagined.
Right.
So what it means for us actually as a scientific community is that we have to be ever vigilant about this because it matters, and I think it's sort of so sort of an interesting question that really touches on what you talked about actually earlier. Can you say something about the changes in connectivity, I think, over time in mammalian brains? Does the decrease in connectivity mean the brains have become simply more efficient, or something else?
So I think that's exactly right. So first, I should say that this is a hypothesis in nervous systems, which is that we know that at some point there are no connections in your brain. At some point in embryological development. So we know that as neurons grow and develop, et cetera, they have to establish connections first. The question is, and the model is that, as far as people could tell, including Huttenlocher, it turns out that neurons produce more connections, meaning if you look at a baby brain, it's likely that every neuron is connected to every other neuron. And that the process of what we call growing up is actually removing all of the connections that are not essentially useful for that brain. So in two particular brains, you might have all neurons connected to all neurons when you start, but in a brain that just grows up in the light, you may get a particular pruning of connections to allow that brain to navigate in the light, whereas a brain that grows up in the dark will still get pruning, but of the connections that weren't necessary for growing in the dark. So each brain ultimately becomes a snowflake, we think, over time, unique. And really what it means, I think, is that the connections that remain are the ones that drive those neurons. And therefore, exactly like the question is asking, the connections that remain become more efficient at driving what happens inside the nervous system.
Yeah, I know, that's sort of, wow, there's a lot of very interesting questions coming in here. I'm going to try and pool some of them to get this right, so that so here's one technology question that we're thinking hard about. Do you believe that you'll be able to reach your goal with existing technology, or will advancements be required?
Well, so again, it's amazing. It's almost like Peter is directing these questions, but I know he's not. So this is something that I'm really fascinated by beyond just neuroscience. It's never, the technology we have at any particular moment, my feeling, is never going to be the technology that we ultimately use to solve any particular problem. I mean that historically, and I'm stealing this analogy from Peter. I think about technology maybe two different ways. If you're in the military and you want to build a battleship, it might take you 5 years to build your battleship. And at the end of those 5 years, you have a battleship. You know exactly what it does. You know exactly its specifications. But that battleship is about 5-year-old technology, maybe 10-year-old technology. Peter says the contrast to that is a place like CERN, where you spend 4 years trying to figure out what is the most efficient technology to get to how you want to map the brain. And then you spend the last year realizing that you've discovered some technology cheaper, faster than you'd imagined to actually solve the problem. Now, I think we have to start somewhere and we have to provide ground truth. So I think the initial wiring diagrams, large wiring diagrams of brains will come from electron microscopy, as I was describing. But I'm not sure that that will be the ultimate technology that we use, for example, to map a human brain.
Good question. I mean, there are several questions, obviously, about disease, which you referred to. I'm going to pick one out, right?
Yes.
But maybe you can, and I'll try and amalgamate it with others. Right. So one question says there's a euphemism that people with autism spectral disorder, ADHD, and other conditions are differently wired.
Yes.
is there a physical basis for that? And do you anticipate finding it in the connectome, or do you think it come from somewhere else?
So it's interesting of the myriad of possible disorders to start talking about. I think autism in particular is one where there is a large sort of proxy data or anecdotal data that suggests that it really is differences in how neurons wire together that causes autism spectrum. In fact, specifically delays in pruning or accelerated pruning over developmental time are what we think are related to autism. Now, I'd like to believe that all mental disorders could be sort of best defined as connectivity disorders, but that's probably not true. Some disorders are probably more chemical, that they relate to things that don't work directly in connections, but chemistry, like adrenaline or dopamine, et cetera, could be responsible. But I think what is interesting is that since right now we have no physical basis for almost any mental disorder, it's likely that many of them will be related to what we call connectopathies.
Yeah, that's an interesting comment. I mean, I say that. I mean, this goes, I think, to your view that you expressed at the beginning, is that we shouldn't have a hypothesis. We should just go measure it.
Yes.
You want to say something more about that that philosophy, which clearly drives you, and why you think it's the right way to do this kind of science?
Yes, I think that's because I'm opinionated. It's not clear that it means I'm right, but I'll try my best, Peter. And again, it's kind of amazing because it's I think a parallel to what's happening. It turns out that in the 1950s and the 1960s, there were philosophers broadly were spending a lot of time thinking about what is science. How do I know whether something is science or something is not science, which has come up recently in the pandemic and how we think about it. And there's sort of two, I think, schools or great philosophers that came out at that point. There's Popper, the philosopher of science Popper, who came up with falsification, and I'll get into this briefly. And there's another philosopher named Kuhn, who wrote The Structure of Scientific Revolutions. And they had two diametric, in my mind, different views of science. The first one is that we should have hypotheses, and we should do everything we can to try to falsify that hypotheses. Meaning we should have a hypothesis that black swans exist, and every day that we collect data about white swans, our confidence in black swans should go down over time. But Kuhn, which you can probably already tell is the one that I am really taken to, says, well, that's not exactly how science progresses. What actually happens, he's the one, I think, to coin the term paradigm shift. In his view, entire fields shift at once. This is the heliocentric model versus the geocentric model in physics, if you will, where all of a sudden people started to realize that everything didn't revolve around the Earth, and perhaps all the planets revolved around the sun. Now, to make those paradigm shifts happen is usually, in Kuhn's world, a piece of data that doesn't fit with the existing model, a surprise, if you will. So that's actually my scientific philosophy. I'm interested in the elements of surprise. The more surprises we can find in our data sets about brains, the more we will be able to get rid of our bad ideas and really push the field forward. So how can you find a surprise? Well, I think I would argue, or I am arguing, that the best way to find a surprise is to collect data that has never been collected before by the nervous system. And it is that simple a logic that has gotten me to connect those.
Good. I'm going to shift you over now. So that, I mean, by the way, there are some spectacular questions coming in here. So we're not going to get all of these. So I want to go into quantum mechanics and quantum computing, which I know is a specialty of yours. So there's really sort of three sets of things which are coming in over the wires. One is, does quantum mechanics affect the brain?
Right.
Don't answer that one yet. Think about it. The second one is indeed noting that Argonne and UChicago is working very hard on developing quantum computers. what impact do you think that quantum computing could have on your ability to solve the problem you're dealing with?
Okay, so I think I'll try the first one because I may have an answer. The second one, I promise, is slightly beyond me, but I'll try anyway. Actually, one of my favorite writers of science writes this book about the quantum basis of consciousness. He's a famous physicist in England. I just forgot his name, Peter. Penrose, Roger Penrose. Thank you so much. And the only thing that I have to say about that is that whatever is the basis of consciousness likely has to happen at body temperature. And I'm no physicist, but I imagine that at body temperature, there's a lot of thermal noise and there's a lot of movement that may or may not mask the effects, the smaller quantum effects that you would see. That's my current theory why I suspect quantum has little to do with brains, but I am talking to one of the experts, so I'll happily defer to you. And the second, I leave that to you, Peter. I struggle to understand in my world, if a quantum computer is going to be 1 billion times faster than a regular computer, then for sure it will help us map the brain at least, some fraction of that faster. But beyond that, I can't think of how specifically it would help.
Good question. So let me come back down to earth. I mean, by the way, I don't think I could answer either of those questions either. They're very good questions. And the reason we sort of work on these problems is that it matters. And that's why it's the kind of stuff we do. So there's something which I think is, I know closer, and you've done this, is somebody who asks, I'm involved in crowdsourced citizen science.
Yes.
Around designing synthetic DNA. Do you envisage a place for crowdsourcing in your quest to map and understand the connections of the brain?
Yeah, not only do I envision it, I think it will be critical. By that I mean, although the algorithms for tracing neurons, the AI algorithms, are getting better and better, it seems unlikely that we won't need humans to error check or participate in that mapping process. In fact, a colleague of ours has made if anyone is interested, there's a crowdsource available called iWire, EYI.WIRE, which is actually trying to crowdsource the connectome of the Drosophila brain. I think that one might be called FlyWire, but close enough. Where the output of algorithms of people who are trying to trace the fly brain are distributed broadly to anybody who can then assist in that effort. And I find it fascinating because not only does it help us, but it increases people's exposure to what we do, which benefits all around.
Yeah, so I'd say that's sort of an interesting one. So let me move a little bit to functionality, right? So you're talking about neuroanatomy. So basically you're an anatomist. There's other things in the brain. There's chemistry, there's stuff going around. So can you infer everything that you need from the connectome, or do we need to do other things? And what else do we need to do?
Yeah, so that's a great question. Of course, I'd like to say yes, so that we could be done. But the obvious answer to me seems, of course not. That getting the wiring diagram is absolutely essential to understanding how the brain works. But just getting the Human Genome Project wasn't completely sufficient to understand the genome. Meaning, yes, we need to know where every gene is. Yes, we need to know how where every connection is. But we also need to know which genes get expressed under which conditions, which genes get downregulated, and certainly in the wiring diagram, which we need to know which neurons get activated and which don't. So I definitely think there will be chemistry, et cetera. I think the way the wiring diagram will be useful is that it will prove that we can account for some things in brains, but not others. And then we'll know where to look, what to look for next to explain some sets of behaviors, but not others. But let me try something also slightly controversial, if that's okay, Peter. I often think about this in the context of the Grand Canal, the Grand Canyon, excuse me, and the Rio Grande. And to me, at some level, the water, the river itself, is the activity, is the flow of information, and the canyon walls are the structure, if you will, of that particular. And like the canyon and like us, it turns out that your activity changes your anatomy, meaning the more you use some neurons than others, it will change their connections. And then the connections change the activity. So you get this loop that goes back and forth until I hope, at least in some respects, the activity and the structure are simply reflections of each other. So those are the things where I think this mapping will help, but it will also help reveal what we don't understand and where else we have to go to look.
Right. So, I mean, that's a great answer. Let me just follow up a little bit on that, because to make it more clear, because there's a really good question here. So, you know, okay, let me read the whole question, because I think it's a good one. Scientists map the human genome. Obviously, there are differences between individuals' genetic sequences that make us different, but we were able to map the basic structure. Will we be able to derive a similar map of neural connections in a way where we can geolocate specific types of thoughts or emotions? Or is brain development individual enough that we'll only be able to map at a more general level?
That's, I think, sorry, I don't know what price tag to put on that, but that's the billion-dollar or trillion-dollar question. Of course, mapping one brain is useful, but it's mostly useful in the sense that it will help us map for cheaper and quicker the second brain and the third brain and the fourth brain. And in fact, I would say the most important question we have to address currently is, what is the variance from one brain up to another brain? So let me repeat the question and pretend like I answered it. The question is, I may have even said this, brains, especially mammalian brains, are snowflakes, are likely snowflakes, each one unique. But even snowflakes have common elements to them. They have 7 arms, or 7 arms that are equally spaced, and some parts that are unique. I think that question of what is the basic structure of the nervous system and what is variable, I don't know how to say other than that's exactly what I'm interested in finding out, and I have no clue what the answer is right now.
Yeah, good question. So let me push you even harder on this. What do you think of the prospects of mapping the connectome of a living animal?
So I don't know. I suspect the connectome, meaning the connections between neurons, I doubt it in the next, et cetera. I haven't heard of anything, a particular technology. Now, we have clever ideas on this, which are mostly cute, I think. There are animals that we work with very rare, occasionally, like the planarium. This is an animal that can regenerate almost any part of its body, including its head. It's a flatworm, et cetera. So we had a project once to sort of remove the worm's head, prepare it for electron microscopy, let the head grow back again, remove that head, put it for electron microscopy, so we could get multiple variations of the same head from the same animal. We could even do it, stop it at different points and get a time lapse. But other than those sorts of cute applications, I can't imagine doing it in a living brain. I think the energy, the resolution required must mean that you require a certain amount of energy to achieve that resolution, which probably living brains aren't compatible with.
Yeah. So I'm pushing a little bit while I'm sort of watching the time here. So, intellectual property. Who owns the IP coming out of this?
Yeah. So if
you've got Google Maps of somebody's brain,
yes.
What are you going to do with it, right?
Yeah, these are fascinating questions. One, I can't wait to be in that position to have to worry about having an entire brain and the intellectual property. My short answer, which I think is just thinking with my academic hat on, is that no one should own this. That this should be as widely available as the genomes are, the original. Now, there will be some genomes currently that aren't widely available because it's their people's genomes and their privacy. So I think we'll have to deal with that when that comes. But sorry, Peter, my first blush reaction is no one should own it.
Yeah, no, that's good. So I say another nice comment, which I think follows on from that. You said your chief motivation is to be surprised. Do you have any fears about what you might find and what it might be useful?
No, I mean, I don't I can't imagine, there's nothing that we could find that's going to humans are going to behave the same way before we found it and after we found it. So I'm not worried that we're going to reveal something deep about human behavior that I wouldn't. In fact, and sometimes people ask me, what if you find that someone is not culpable for what they do? Meaning it's all in my circuits, so it's not my fault. My neurons made me do it. And I do think that's an interesting discussion around what our kinds of studies mean for free will, for culpability, et cetera. But I used to worry about that, but I don't worry about that anymore. Because I think as society, we have some experience in this already, meaning we already know that there's a physical basis for a lot of these things. The example that I use is that if I got arrested for driving under the influence, 40 years ago, people would assume that I lacked moral character or I didn't have self-discipline, and that punishment would be a way to deal with me. But now we realize that there are diseases of alcoholism that have genetic basis. And now if you get, perhaps if you get arrested or charged with a DUI, the emphasis is on therapy, because we realize that that's a better way to resolve the problem. So I used to worry that brain maps would take away free will and culpability, but I realize it's sort of happening already with what we know.
So, we're at time and I'm missing all kinds of questions. I've got one last one that I'm going to sneak in, which is, does the university have any means of generating practical applications from the kind of research you do?
Does the university, I assume yes. This is a version that I could think of, if this falls into the category of practical applications, is this idea of how we can apply this technology to patients already. Maybe that's a practical application. How we can work really closely with surgeons and physicians at the university and do this large volume nanoscale mapping on biopsies, on mini brain, many other things that we could talk about, so that we could improve the ability of physicians to predict what will happen to patients, and ultimately improve treatment as well.
Thanks, Bobby. I mean, we're really over time. I apologize. There are several questions which are unanswered. I promise everybody that we will actually address these offline, because there are a whole bunch of really good questions that I would have liked to have skewered Bobby with. But I do want to say, firstly, thank you, Bobby, for a great presentation for this really exciting frontier. Actually, I think that we're engaged in at UChicago. It's just incredibly exciting. And I will say, I don't think we could do this anywhere else. UChicago is the place to do this. It's sort of special. So I will now turn it back to Andrea. And I suspect the problem with these Zoom meetings is that we die unpleasantly. I do want to thank the whole audience for coming.
Yes, please.
Tremendous number of people who came in this. Lots of questions here. Don't hesitate to contact us after this. We're very interested in your views. It's sort of interesting to see that the depth of the questions that get asked, they're very stimulating. They change the way we do science. I mean, you may not realize this, but it does, because these kind of questions, particularly in these frontier areas, are really important. And it reminds me of how much I enjoy being at the University of Chicago and what an exciting place it is. And thank you for being an alumnus.
Thank you.
Thank you both so much. Thank you to Professor Kasthuri for sharing your research and your knowledge with us, and to Professor Littlewood for facilitating the conversation. Thank you all for submitting your questions, and I'm sorry that we couldn't get to all of them. If you'd like to hear more from Professor Kasthuri, listen to the Big Brains podcast from the University of Chicago. He was one of the inaugural guests, and he is featured on episode 12. You can find that episode and others on bigbrains.uchicago.edu. Thank you again. We'll see you soon.